Hello, and welcome to Cognitum, a show dedicated to exploring the present and future of science and technology. I'm your host, Iosif Gershtyn. Tonight, joining us is Walter Patterson, a biotechnologist and founder of MiniCircle. His primary goal is the development of gene therapies to negate the process of aging. In 2017, Walter and his business partner, Mac Davis, set out to design a novel gene therapy vector platform able to deliver a wide variety of targets into human cells. The primary focus of which were proteins which could potentially treat age-related illnesses such as sarcopenia. Today, such work has been demonstrated in clinical trials on the Caribbean island of Roatan, whose publication is currently within the review process. Walter's academic work has been published in peer-reviewed journals, and his current work has been featured in several documentaries and news publications. Walter, welcome.
Pleasure to be here.
Walter, tell me about the clinical trial you are running.
So, it was comprised of 42 individuals of mixed sex, age ranges from 23 to 89. So it's a fairly comprehensive study in terms of the entirety of the population, and we didn't run a control on it. What we did is we got prior data, so these included measurements such as the serum levels of follistatin, the levels of certain blood markers such as testosterone, LH, follicle-stimulating hormone, etc. We performed DEXA examinations, so essentially low-grade X-rays to actually see the density and the composition of the human body. And additionally, we also ran immunoassays, such as ELISAs, on the actual blood serum back in the United States. And so from these before bits of information, we were then able to administer the therapy and then take points afterwards, the administration. It was a kind of decentralized trial in terms of patients weren't required to stay down on the island for the full duration of the experiment, which consisted of a 3-month primary observation window. They could go to their doctors, etc., in the United States or wherever they were from, and we could still get the blood data and also the DEXA examinations in order to establish if there was an effect based on the therapy.
And what were the results?
So the results, based on all the data we've accumulated and got back, we saw increases in across the board, generally increases in muscle over the 3-month period, based on the DEXA examinations of about 2 pounds on average. Some in the trial, we could definitely see much more of a difference. We had one patient who was quite the exception at 15 pounds in the 3-month period of trial. But keep in mind, it's a very large age range. So the 82 pounds for an 89-year-old is still remarkable, just because they tend to be less active. Now, in terms of body fat composition, we saw about, on average, about 1% or 0.8% to be exact, in terms of average fat reduction. And we also saw epigenetic changes. We used a third-party tester, True Diagnostic, and we were able to see intrinsic and extrinsic age. That's based on a calculation based on methylation, such as Dunedin pace, or as well as certain other blood markers and certain other factors, such as white blood cell count, etc. So based on those numbers, we saw intrinsic epigenetic age of a reduction of about 3 years on average, and we saw a 7-year reduction on average, the mean. In addition to all this, we were able to see a typical increase of the follistatin levels in serum based on immunoassay by about 15 nanograms per milliliter. So this is above what an individual would typically see as an increase in range over the same 3-month period.
And what is the therapy that you were trialing?
So the therapy consists of a plasmid vector system. So plasmid vectors have been used for decades now for, transgenic modification. This is all the way from vaccines to actual therapeutics. Now, the issue and what we've overcome essentially in our particular vector design is the issue of transgene silencing. And we've done so in a manner that essentially hides away certain issues, certain sequences that cause transgene silencing to be more likely to happen, such as the origin replication, etc. It's typically bacterial components needed for the production of the plasmid. Now we take that plasmid and we take a cationic polymer, cation just meaning positively charged, and we use that as a transfection reagent. That transfection reagent complexes with the DNA, allowing then the DNA to be uptook into the cell. Once inside the cell, we have helper sequences that eventually get it into the nucleus. Once in the nucleus, it will code just like any other bit of DNA.
And what is it coding for?
So this particular trial, it was coding for follistatin 344. Follistatin 344 is a naturally occurring hormone-like protein. You have it, I have it, the dogs outside probably also have, or definitely have it. And it's a very conserved protein that has a couple different isoforms, but its primary activity is as a selective myostatin inhibitor. So are you familiar with the idea if you don't exercise, your muscles begin to atrophy?
I've heard of something like that.
Yes, and so we also see something akin to this in astronauts, but also this happens particularly as people become less active and also during aging. This is typically in reference to aging is called sarcopenia. I'm sure you've had your grandparents or older relatives, as they get older, they're not able to function or do as much physically. This is a characteristic of it. By selectively inhibiting myostatin, which is essentially the receptor is responsible for initiating the breakdown of muscle, you're able to essentially tell the body, don't break down the muscle. And so that's one of the mechanisms primarily found for follistatin.
There seems to be some studies that indicate that elevated levels of follistatin have harmful side effects. How do you approach this issue?
So, for example, there have been several papers on one disease class or another. For the one off the top of my head, diabetes. These are essentially studies looking at large groups of individuals, looking at what any kind of marker that might predict under a typical regime of any particular protein, what have you, that would indicate a potential progression towards disease. Now, that doesn't necessarily mean mechanistically that the follistatin caused the disease. For example, if I have some kind of muscle cancer, one could hypothesize very well that follistatin increases not necessarily due to the actual cause of the cancer, but rather just a symptom from the actual disease itself, or the body's alternative way of mitigating the damage based on underlying health conditions.
So you're saying that the elevation of follistatin is not causative, or it's just an association?
It's just an association. There's no direct mechanistic evidence in any of these disease models that would indicate that follistatin is a culprit.
And so after the results that you've achieved in this study, what are the next steps that you are envisioning?
So based on the trial results in which there were no negative clinical outcomes, we are going to proceed along the same pathway that you you typically see in any kind of drug discovery. So phase one is essentially determining whether something is toxic or there could be complications. Phase two, the primary goal there is to determine dose and begin the process of determining efficacy. And then finally, phase three, determining the efficacy on a particular disease target. So as we approach this milestone of actually starting to begin a phase two, we're looking at several countries around the world, including Canada, Mexico, and again, the Special Economic Zone in Roatan, in order to proceed forward with further experimentation.
So outside of this particular therapy, what is your vision of your company, MiniCircle, going forward?
So, as established before, this is not particular to any one gene. We do have our limitations with our vector system, mainly that we're a localized vector system. It doesn't spread throughout the body. Typically, when we transfect, it is at a localized site via injection, and typically those cells don't move anywhere. And it's only at that spot of injection where the transfection is able to take place that you actually see the genes get into those cells. So in that way, we're relegated to either small systems, maybe things like eyes, what have you, but mostly we're relegated towards excreted protein. So protein that comes from a cell out into the bloodstream and then circulates throughout the bloodstream. But that still envelops a wide variety of potential. So we can treat things, we have several different proteins and targets we're going to be looking at proceeding just this year to phase 1, such as luteinizing hormone, potentially things such as later down the lines, such as certain kidney markers and kidney proteins. And then later on, that we're still in the in vitro and animal trials, things such as VEGF, things such as cellular reprogramming, which is still for us in vitro stages to make sure the tox and everything is good. But in terms of the vision of the company, I guess, we want to be essentially the first line of gene therapy, something that's not good or particular in a very niche use case, but something that can be generalized to a lot of diseases. And we're open to even the idea of licensing out the vector system so other people can develop along the same lines that we have.
So you have a platform company.
Essentially.
So tell me a bit more about this platform.
So, as I stated earlier, the platform is a circular bit of DNA, plasmid. Over the decades, there's been several attempts to make plasmids functional. For example, if you take a regular plasmid, such as PUC19, you code it up to be a mammalian vector, so you have mammalian promoter, mammalian transgene, all the stops. You'll still only get about maybe 1 to 2 days of expression if you're lucky. And this is because of transgene silencing. Now, it can occur for a wide variety of reasons, both sequentially and as well as what the actual protein you're actually using in some cases. Is it of bacterial origin? Is it something that is homologous to the animal or individual you're already testing on? But essentially what we've done is we've mitigated some of those sequences through a variety of little techniques where we've actually gone in and engineered intronic regions and other components that help the actual expression last for much longer, because you don't end up having that transgene silencing take place. So we've worked very hard to mitigate that as much as we can. And the benefit of this is we can actually induce transgene silencing. So in the same way that a plasmid would express for maybe 1 or 2 days under a standard regime, we actually on some of our vector systems have a kill switch. This kill switch is typically induced by an outside small molecule, namely things such as potentially cumate or tetracycline. So you can administer either this into let's say I have a petri dish of cells, I can administer that dosage in, and I can watch whatever transgene I'm looking for to stop the expression of. So one example that stands out in my mind is we've done a lot of work with GFP, the standard glowy. And
GFP, could you...
GFP is green fluorescent protein. We particularly like it because it's you can take an optical measurement. You can literally see it glow. So for us, it becomes an inexpensive way. Instead of having to chase down an invisible protein, we can actually just have the protein quite easily visible to us. So when we're doing a lot of these in vitro experiments, where we're actually testing out vector designs, etc., what we've done is we've typically gone with GFP or a similar protein that expresses light. That way we can actually test very cheaply, quickly, a bunch of different iterations without having to resort to Western blots or ELISAs to confirm that the protein has stopped expression or not.
So by learning how to silence the transgenes that interrupt long-term expression of the protein of interest from the plasmid, where do you want to take this platform?
Well, as I said before, in-house have a bunch of essentially demonstrations that the possibilities of what our system can do. And we're very much open towards the idea of collaboration and licensing with outside parties who, let's say, if you're a laboratory here in Boston and you have a particular protein that you think is the bee's knees, and a system like ours might be amenable to it, where it has some kind of effect or has the potential of some kind of effect, where it can be in the circulatory system and still be fairly useful. And if you would like that to express for a generative, a transient period of, let's say, a year or two, then we might be amenable to it. And so you can license our technology, put in your sequence, and we can even assist in terms of having a pipeline, such as the Special Economic Zone, as well as some of our partners and CROs, essentially a pathway towards regulatory approval in various countries.
Tell me a bit more about this kill switch. How does it work?
So the kill switch, as I said before, are you familiar with the intron and extron?
Tell me.
So basically, the best example I can give is, you know how like my skin cells and my brain cells have the same genetic code. Part of the reason they express different things is due to a wide varieties of factors such as methylation, but also on top of that, some of that code isn't actually used. This is called intronic, so it's inside the genetic code. It doesn't come out, even though it can code. So it's got sequences that would be a protein, but for whatever reason, it's not going to code. And then you have extrons, which are sequences which actually do express and do code. So in that way, we've actually been able to go in and engineered varying flanking regions, so on the sides of a particular transgene or on the sides of the intron. And we've actually been able to go in and code up those introns and extrons regions and essentially almost make like a circuit or an ability to respond to it. So let's say, we have operators essentially on that allow it to be coded and allow it to be seen by your internal genetic, machinery under the event that we want it to be like, hey, look at this sequence right here. Hey, and of course your internal cellular machinery is going to look at that sequence and be like, hey, I don't want that coding, whatever that is. So we can actually not only have those bacterial bits in there and be hidden, we can actually have them pop up and show in order for the transgene silencing to occur. Does that make any sense?
What evidence do you have that this kill switch works?
As I said before, so when we were doing a lot of this iteration, we went back to the lab bench, as it were. So one of the things I really like is using human cell lines. And I love culturing in vitro human cells. So I like being able to have a system that is very analogous to humans, while at the same time I can have multiple iterations. And as I was saying before with the GFP, I know that human cells, I don't know if you know this, they don't typically glow under blue light. So that's a really good indication that I've done something and put in a transgene that wasn't there before that is there now. And so I can take and I can look at those cells and I can dope in the particular kill switch, the actual chemical that would essentially make those components visible, and induce transgene silencing. I can watch those cells go from a nice, eerie, kind of green color, glowing in the dark, to nominal state. So back to not expressing, that GFP.
Outside of the model of the green fluorescent protein in vitro, do you have in vivo validation of this?
We're concluding currently a trial where we've given people the follistatin, we've given them the vector, and we've seen the expression in their blood, and then we've administered the kill switch to them. And we've seen a drop in the level of expression based on the ELISAs. So sandwich types ELISAs is a type of immunoassays. It works based on antibodies, and we can use that system to particularly identify what the concentration is of follistatin or any other transgene. So we've actually been able to take humans, we've been able to administer the therapy to it, then we've been able to have those volunteers take a kill switch, then we've had those volunteers run another ELISA on them, and we've been able to see the levels in their blood drop. Now, we've not only done that, we've also continued follow-up with those individuals for 3 months, looking at their blood to make sure it doesn't just rise back up or something other, weird happens. So we've seen the drop, we've seen it maintain that drop.
And the level at which maintains, is that equal to the level that it was before the follistatin therapy?
Yes.
Do you have a way to titrate the amount of follistatin that the therapy produces?
In terms of determining the concentration per dose of the therapy?
Yes.
So individuals will express variable amounts. This is something we're still trying to determine an exact, per microgram of, vector system to nanogram per milliliter of follistatin expressed. There does appear to be some variance within individuals. We have not been able to fully parse it out conclusively yet. Typically, though, we are able to establish a logistics curve of, on an individual level, of how much a particular amount of therapy should express in an individual. But outside the individual, it's difficult to titrate as of yet, and certain factors seem to be relatively unknown, such as what's the concentration of your myostatin receptors, what's the concentration of activin. All of these components do play an end result on the actual concentration of the follistatin at the end of the day in one's serum level. Due to its short half-life, being only approximately 90 minutes, you have to have a constant production and supply of it. It doesn't stick around for a long time. So these factors that are taking away from the follistatin in your circulatory system will also affect how much we actually see at the end of the day in the transgene.
Are you worried at all about increased upregulation of myostatin receptors in muscle cells?
At current, we haven't seen that. We haven't seen upregulation in terms of, particularly in mice. It's something I think we're very cognizant of. It's something I think we are pursuing, particularly with future tox studies, to make sure we have no upregulation, and if there is, to what extent, which would indicate, to us how many years it would appear that a person can be on or off the therapy.
So, in an ideal scenario, where do you think that gene therapies, including your vector system, Minicircle's vector system, and others could take us?
I think, ideally, a better tomorrow. For example, I wear glasses. At one point in history, it was I would just be half blind and could barely see. Everything would be blurry. But using glasses, I can see. My father has type 2 diabetes. Without insulin, he would die. This is the kind of possibility of providing a better life and a longer life through these advents in technology and science. So for me, I would like to see gene therapy be fairly common, fairly widespread, and not everything needs to be very invasive. You know, for example, I think that there's a potential future in which, you know, gene therapy may be an outpatient procedure in the same way that someone would go get, you know, their braces done. Some people might want to go get a particular gene therapy to alleviate some issue in their life or to enhance their already existing capabilities, whether that be their strength, how long they're expected to be able to work in the workforce, things such as even down to intelligence or other matters. I foresee a future where people have an active choice in who they are and what kind of ability they can have in terms of, you know, their capabilities. And I believe in my whole heart that, you know, this is an avenue by which people get a choice in what their lives ultimately be. You and I are both dealt a hand at birth, genetically. And some people really got a bad hand, really bad. And so with technologies like this, in the same way that I have to wear glasses, or that some people require braces, it's going to alleviate that suffering.
So getting back to the technical side of things a little bit, how do you contrast the advantages and disadvantages of your plasmid-based platform with some popular adeno-associated virus platforms or messenger RNA platforms?
Absolutely. So in my view, all these are different toolboxes. So for example, if I want something to express for a short time, let's say like in a vaccine, I'm going to reach for something that expresses for a short time that I can reliably know that it's not going to continue on and cause potential immune issues. So I'm going to reach for mRNA or canonical plasmids, such as we see in the COVID vaccinations in the United States, as well as in the example for plasmids, certain vaccines in India. Now, if I want something to express for a transitory period of time, then I would reach for something such as our vector system, which, you know, I don't want it necessarily integrating into my already existing chromosome. I just want it episomal, and I want the knowledge that this is essentially, I'm on almost like a trial period. I'm on a period of time where this will be expressing. If I want to essentially re-up my dose, I absolutely can do that after a certain period of time. If I need something that needs to be permanent and it needs to be integrated, let's say I have some issue with a particular protein in myself, then I would reach for the adeno-associated virus and I would reach for CRISPR. You know, CRISPR is extremely good at taking particular things away. You know, its whole major purpose is to cut things in and out of particular sequences. And it's really good at that job. Fantastic even. An adeno-associated virus is a beautiful example of being able to deliver a particular payload and integrating into a chromosome. Now, in the case that there's something messed up or something completely absent that I would like there permanently, I'll reach for those. And so each of these different tools gives us the ability to provide therapies and opportunities where opportunities didn't exist for the misfortunes.
So you believe that these various different platforms for gene therapy will all have specific use cases and could all potentially bring therapeutics that would be useful down the line?
I absolutely believe that. I think that, you know, if I need to go unscrew, you know, if I need to use a particular tool, let's say if flathead versus Phillips head, you know, or a particular kind of nail, I'm going to pick the nail that is good for that thing. If I need to nail something into a block of granite, I'm going to use a masonry nail. If I need to nail some wood together, I'm going to use a carpentry nail. If I need just to put up some wallpaper or something very light, I'll use a finishing nail. All these are different types of nails, and they're putting two things together by being hammered in, but each one has its use case. In the same way, I don't think there's going to be a one-all, be-all, genetic therapy solution. In the same way that there's not one in chemistry. There's no panacea for anything. So it's going to be the case that you're going to pick the tool for the job. And some tools aren't suited for particular jobs, but are suited for others.
Did you set out to create this plasmid platform, or was there a certain breakthrough or discovery that got you to develop this platform?
Yeah, we were introduced to this even concept and technology by various colleagues of ours. And we were introduced, for example, we were introduced to the particular even transgene. And we saw a great potential here. Now, at the time, there was several issues with what some of the vector systems that our colleagues had shared with us and showed us. And this has been the case for a lot of literature as well. So what we did is we set out to design the most scalable in terms of being able to make hundreds of thousands of liters of this material fairly quickly with already existing infrastructure. It needed to be relatively acclimatable for most situations, so it needed to be able to express in a variety of tissue types fairly reliably, and it needed to be relatively long-term in its expression. So we were, some of the first vector systems we were working on barely had a few months of expression time, and so over several iterations, we were able to come to the final vector system which we have today. Internally, we have vector A through X, and then we just started numbering them because X sounds cool. But in terms of, all science is built on the shoulders of giants. Plasmids have been around for decades. Follistatin has been around for multiple decades. It was all the way back in last century when it was discovered. And it is a thing of, I am extremely grateful for the scientific community and the literature provided that's allowed us to, develop this further on than anyone else has. And, through the iterations and through multiple in vivo and in vitro experiments, we've been able to determine a final vector system that seems applicable for most situations.
So Walter, one of the differentiators of MiniCircle, at least in the public eye, is how you've gone about pursuing your clinical trials. Could you tell us about that?
Yeah, so we just concluded one in a special economic zone in Honduras, off its coast on the island of Roatan. So on that island is a special economic zone. Essentially, the Spanish is ZA. That's the acronym for it. It stands for Zone for Employment and Economic Development. And it's essentially another municipality with specialized laws. Now, what these laws allow is essentially, let's say if I want to come in and build a house, well, I can choose to operate as a construction company under different legal regimes that, as long as they meet the standards of Honduras. So I can say, I'm going to build a house under the American regime. I'm going to build a house under German regime. I'm going to build a house under Israeli or what have you. There's a particular list of countries that you can pick and choose from. And as long as you either, you have to build it according to those particular sets of laws. There's also the option to pursue under common law. And common law is essentially saying that if I hurt someone, then I am able to be sued for 3x the damages, and that everything that I do in terms of my business is liable for that, and I have to have insurance to essentially mitigate that liability. Now, there's a couple different examples of particularly regulated industry, if you do pursue the common law, such as medicine and construction, typically industries where people can get hurt fairly easily. So because of that, you can operate a clinical trial down there under review by a body of doctors, typically called an IRB, or Investigational Review Board. So this is essentially a ethics and scientific council of particularly medical doctors that meet and debate, and also have to, double check everything if you have a proposed procedure, and have a doctor there who's willing to administrate. So essentially acts as a double check and regulatory body on top of the insurance that would be required for such a trial under almost most regimes. So for example, the clinical trial we've done, the review body, we went through an IRB, and so there was a team of doctors and scientists who acted essentially as a third party, and we proposed with the medical team down there in Honduras, we proposed a particular protocol. And so we had the ELISAs, the DEXAs, everything was written out, everything was prepared, and then the IRB then presided over and determined whether it was legitimate, and whether, for example, like the QCs, what would be followed, what would be essentially exclusion criteria, the whole nine. So essentially what a governmental body in any country, such as the FDA or the Department of Health of one country or another, would do. However, this is private, so but is beholden to, the same kind of standards due to the liability that would incur due to the common law. So, one thing that I always found funny is we were constantly audited by the insurance company, because they did not want to insure anyone that wasn't following to the letter of the law. So, you can see how a financial incentive can motivate to make sure everyone is following the parameters by which the doctors and the scientists on the review board had prescribed.
So this provided you an avenue that was less expensive and more expedient than a large country's Department of Health, Ministry of Health, Food and Drug Administration, etc., other governing bodies that would oversee such clinical trials in general.
Yes, particularly with... Now, that being said, we still had to provide mice data. We still had to provide in vitro data. We still had to provide, even under this kind of regime, even case report can sometimes be required, where only a single or a couple individuals are administered, particularly in approachment of phase 1, before it's tried on a larger population. So, keep that in mind, that you still have to provide that level of evidence that you would under any kind of regime, tox reports from animal models, and actually looking at cells. What is it doing genetically? Are we seeing, markers that might be in association with cancer? What's the morphology of the cell look like? Is something going weird here? And so you still have to provide that same level of evidence. However, there is a benefit of it where it is more expedient. And, particularly in the United States, I have no issue with the FDA. I have nothing against them, but I'm sure several of our colleagues could, state that it sometimes takes a while, and they tend to be fairly slow about IND and getting it into trial.
What excites you the most over the next 3 years with gene therapy?
Honestly, the thing that excites me the most, you know, from my perspective, going from a company of a few individuals to now we have a team of doctors, both PhDs and MDs, getting to go to the office and seeing the company grow really excites me. Seeing burgeoning ideas on new places we can take the vector system, new additions we can have in terms of both transgene target as well as control over our expression. We currently have this vector system which has been through years of iteration, but it may not be applicable in all cases. So I'm really excited where we can take our tech, not just where it is right now, and apply it to multiple different things. I'm excited to see what we can develop in the future. I'm also excited for collaboration, both internally and externally, with more and more new people getting into this field every day. You know, every time I go to a conference, I'm terribly excited to see what discoveries are being made. They dropped a new organelle last year. Like, these are all, every day something new is coming out. And the idea that, you know, death and disease could one day be simply an option, it's terribly exciting. You know, 3 years ago, our vector system didn't have a kill switch. Our system was operating on multiple complicated purification steps, multitudes of different types of entangling and disentangling enzyme systems, whether that be, you know, and as well as the fact we were still getting issues with transgene silencing, even on our most, you know, our best performing systems. And now we've mitigated almost all of those issues. So it's interesting, and the only thing I know is tomorrow will be a better day.
I hope so. Thank you for joining us tonight. We hope you join us next time as we examine and elucidate the frontiers of science and technology, with the thinkers creating our tomorrow. Until next time, farewell.